Hello and welcome to another episode of the Omics Cast. And today I'm going to talk about cell-free DNA, ChIP-seq, and cell of origin. I'm going to talk about a very recent preprint on the bioRxiv entitled ChIP-seq of plasma cell-free nucleosomes identify cell-free cell of origin gene expression programs. And it's a paper written by a bunch of people in Israel. And it's along the line of some other episodes I've covered along liquid biopsies. So what can we get from cell-free DNA from a liquid biopsy, from extracting a blood sample from someone? In the abstract, they say they basically use blood cell-free DNA, cfDNA, and that is derived from fragment chromatin in dying cells, and then pull down the histone marks, the covalent modifications in the histones that are wrapped around double stranded molecules of DNA in nucleosomes, and they generated these resequencing libraries from low volumes of blood, and then looked at where those chromatin marks associated with active transcription factors where they laid on the genome and what information they provided around tissue of origin, cells of origin, and their ability to predict disease or disease conditions. So the way they did it was by taking plasma from healthy individuals. And they took about a mil of plasma, which they say contains the equivalent of about 1,000 genomes. So a mil of plasma is really very little. You can get 1 or 2 mils of plasma from 10 mils of blood, and tells me 10 mils of blood is the size of a lipstick. It's not that much amount of blood. And so in that much amount of blood, there's about 1,000 genomes worth of cell-free DNA. And they said these sometimes you see a marked increase in the amount of DNA in certain pathologies like cancer, certain physiological conditions like exercise, or exercise actually bursts some cells into in our body, and the DNA of those cells goes into the bloodstream. And then they say these cfDNA fragments are quite short-lived. They have about the half-life of less than 1 hour in our bloodstream. And then they say, what can we do when we look at this DNA? They say the precise genomic location of specific cfDNA samples reflects their nucleosome position where they fall in the genome, which they say allows us to identify the source tissue and then suggest the cfDNA cellular origin. Now, this is a bit of a schematic of how they did it. So their argument is our bloodstream contains DNA that comes from either the blood cells themselves or from different organs in our body, here highlighted the brain or the heart or the liver, etc. And then these have, if you want, the nuclei from these different organs at different genomic positions have different colors here. So a color for heart, a color for liver, etc., etc. They'll call it something slightly different later on. But in a color analogy, they have these nucleosomes in different colors that have different marks in the histones. These are covalent changes in the histone protein. And they can pull them down with different antibodies and see where the reads accumulate, right? So you use an antibody to bind the different histones and then sequence the DNA that wraps around it with next generation sequencing and find the different peaks of coverage and see where they map in the genomic coordinates. And so that's a little bit how they do it, and they'll talk about the details of how to pull them down, where do they attach, and where do they do the library, etc. We'll cover it later. And so their reasoning is if active chromatin marks are retained on circulating CF nucleosomes, then by capturing these marks on the nucleosomes will tell us how the different transcriptional activities happen within different cells, right? And what they show is that they can specifically capture nucleosomes with different types of the marks, and then they can recapitulate their associated gene expression from these marks. So they're going to talk about which marks they did. Here's a representation of them. H3K1, K4ME1, ME2, ME3, and K30C ME3, which fall into different places in the genome, and see how for two different individual plasma samples, they're quite reproducible. And not only that, they also are reproducible against the cell-based ChIP-seq, right? And here in different colors, you can see. They're reproducible but not identical. And then so the changes between those, they argue, reflect the changes in cell specificity, tissue specificity. And so the genes here on top, either marks being at the promoter or the gene body, etc., they will tell us about the biology of what's happening to those genes. If we turn this around and represent the genes from the TSS onwards, or an enhancer region in the middle of an intergenic region, where do these different marks that they did fall in? Then H3K4 trimethylation is TSS associated, promoter associated. H3K4 ME2 is before and after the TSS, but is silent enhancers. Productivity ME1 is along the gene bodies and marked enhancers, and K36ME3 for silent genes. And so what we can see is that they have these 4 marks for each of the samples, and then they can compare it to, well, they can evaluate if this makes sense, if this makes sense to existing data that happened. Can we do cell-free ChIP-seq? They compare it to the roadmap of epigenomics data sets, and they see a remarkable agreement in the content, in the peak content in Genome Browser. And so that's a box ticked. What could be the other concerns to make you wonder if this is working or not? When one is that the contamination, the DNA here comes not from the cell-free DNA, but contamination from white blood cells, from the tube lysis, the in-tube lysis. And they say they think that not much of this is happening because they can control for the fragment size distributions. Given that the mono and dinucleosomes of the cell-free DNA have very consistent fragment sizes, then if they see fragment sizes different, that they can tell that they come from different places, right? And then they also said that they identified hundreds of promoters carrying specific marks that we know from having done the biology before, are absent from white blood cells, right? So they can gather from this what's the proportion of cfDNA marks or reads compared to any possible contaminants from any of the cells bursting in lysis. And they say this makes them confident that this is working. And then they went on to look at healthy but also non-healthy individuals, right? They did 15 healthy subjects, different ages, and then they took samples from emergency rooms from people that had their heart basically exploding, acute myocardial infarction. So lots of cardiomyocytes bursting and shedding DNA into the bloodstream. They also looked at people with stomach cancer, gastrointestinal cancer, and people with partial hepatectomy, people that got a serious lesion to their liver. And then so the rationale goes, all these are potential cases where there is shedding of the DNA that doesn't come from the normal places, right? And they realize that the people with AMI have an increased amount of cfDNA that's consistent, and that it comes through cardiomyocytes. And they're going to show some of these examples graphically later. They do say, though, that for most of the samples, for all of them, there's always a large group of genes that are sort of stable, housekeeping. They're always consistent. They have the same marks for these 4 histone marks, and there isn't much going on with them. Now, there's about 10,000 genes. How about the rest of the genes? Well, there's a group of about 8,300 or so that show a lot of pattern, self-specificity patterns, right? And they compare this to the GTEx compendium and the Blueprint epigenetics project, either for gene expression or from other regulatory genomics data sets. And they say Based on this, we think we can cluster this into what they call putative clusters, right? With doing the pattern comparison with enrichment analysis. And so here is a graphical representation of that. They say there's a group of genes of about 9,000 where things are relatively constant. There isn't ups and downs between the different conditions and also what we see in the reference data set, roadmap, etc. But then there's a cluster that varies, and it varies quite a lot. It varies by condition, etc., etc. So let's concentrate about and looking at this cluster of changing this set of changing genes. And then so for this set here, they look at the differences in clustering them and see what they see. They see clusters of cfDNA. They see clusters in roadmap, in GTX, and in blueprint. And then they see clusters that are essentially tissue specificity or tissue type or cell type, right? So they see a liver cluster, they see a neutrophil cluster, they see a megakaryocyte cluster, neutrophils and monocytes, astrocytes, fibroblasts. A lot of it are blood cells. Why? Because, well, in the bloodstream, there's a lot of DNA that comes from blood cells. But they also see clusters for other tissues. And because they did gastrointestinal patients and they did patients that had heart problems, and they did patients that had a liver cut in part, then they can see these clusters coming together very neatly, right? And so they see this from a naive clustering, basically, and just then coloring it and comparing it against the reference data sets. In terms of proportions of how much of this comes from different places, neutrophils and monocytes are a bunch of the big portion of it. NKs and T cells and B cells also represented. And then for the different other tissues, liver is up there, and then brain and heart are the next two. Even though, you can think, well, brain, we have a big barrier between our brain cells and the rest of the body in the bloodstream, but they can see brain present in the bloodstream and also heart. And so they can see these different clusters in different samples, some for the healthy, some for the patients, and the patients show up as they expected for different conditions. Examples of this over time, the people that had their liver chopped off goes from high until after a few hours it recovers and basically says, okay, the liver has recovered now, it's not shedding DNA massive amounts anymore, etc. Heart is a little bit like the other way around. You have a heart attack or some condition that kills your heart cells, and over a period of several hours, you see more and more cells being shed into the bloodstream, right? And so the progression of different conditions here is quite remarkable. For the AMI here, for the epithectomy, it's a burst and then it goes down, etc. And then if you look at this by the cluster you get from the different cells, there's something that remains quite common about them. Monocytes, neutrophils, most of them have it, and including liver. We all shed a little bit of DNA in our liver because liver is a highly vascularized organ, but also is an organ that is essentially filtering everything that happens in our blood, right? So it's there to keep things in check. And so it's normal that we constantly sacrifice liver cells for the benefit of the survival of the organism. If you look at this in classes, but in specific gene clusters or gene locations, you see how here in different colors for different marks, you see different classes of up in the promoter, up in the gene body, up in the promoter, not in the gene body, etc., right? And they say they can give letters to these different types and class them, and then classify how much of an overlap is there between those on a gene per gene basis, if you want, right? And here's an example of combining these two ways, combinations of when is the different combination of marks up and down versus where do they fall in the genome, and then the layer of the tissue of origin, or the condition of the organ of the individual. So a very clever way of combining 4 different marks and deconvolving what the cell type is doing, right? This is a small scale project if you compare it to what ENCO did a few years ago, what the roadmap of epigenomics did, projects like this did. But it's clever in the way that they concentrated on finding this signal in cfDNA, not in specific cell types or tissues, in cfDNA. So as a summary, they basically took from plasma all these little nucleosomes with a little bit of DNA wrapped around, each with different marks in the histones, covalently marked in the histones, different colors here. They did 4 different antibody pull downs. And then after that, you release the molecules, do the library preparation and sequencing, and then do all the clever bioinformatics to map it up, right? So very interesting paper. I hope you enjoyed it. It's more of these kind of papers in the interface, let's say, between liquid biopsy, next-generation sequencing, regulatory genomics. I hope you enjoyed this episode. I enjoyed doing it, and I hope you look at the other episodes where I've done other highlights of other papers related to liquid biopsy, next-generation sequencing, what it can bring for diagnosis of disease. Please follow me on Twitter if you may, twitter.com/albertvilella. I'm happy for you also to contact me, connect with me via LinkedIn, and contact me for any questions you may have. I'll try to answer all of them. And this slide deck is available upon request. I'm quite liking these episodes that I've done. I've done a few of them, and I think YouTube is quite a convenient way of doing it. The feedback that I get is from people that watch the YouTube videos, that subscribe to the channel, that post comments about the videos on Twitter, that contact me on LinkedIn saying thank you for having mentioned this in your videocast. So please keep doing it, because it gives me and encourages me in doing more of this. I'm quite happy to continue doing them, seeing the response from people online and social media. Thank you very much, and I'll see you in the next episode.